What are enzymes? Globular proteins that catalyze reactions, accelerating reactions by factors of at least 1,000,000. What is the fastest known enzyme? How fast is it? Carbonic anhydrase, hydrates 10 to the 5th molecules of CO2 per second, 10 to the 7th times faster than uncatalyzed reaction. Enzymes are blank. What do they lower and where do they only work for? Highly specific, lowers activation energy, cannot make a reaction happen if it is unfavorable. What enzyme hydrolyzes lactose? Lactase. Rate of reaction is influenced by concentration of substrate, concentration of enzyme, temperature, pH, presence of inhibitors. What do enzymes do in veggies? They help it grow and mature. What happens with the enzymes when you freeze veggies? Enzymes slow down when freezing, but will not stop. What will deactivate the enzymes in veggies? Blanching denatures enzymes and stops enzyme activity. What is blanching? What is the process? Mild heat treatment used to inactivate enzymes before freezing. Expose product to hot water or steam between 80 to 100 degrees C for a short time. Once finished, quench in an ice water bath to prevent further cooking. What is important for blanching, in terms of control? Control of time and temperature. What happens when you underblanch? All the enzymes do not get deactivated. What happens when you overblanch? Cooks food and introduce losses to tissue damage. How must product be for blanching to work? Product must be in equal pieces and as uniform as possible. What is peroxidase? More heat-resistant than other enzymes in veggies, works to remove peroxide and convert it to water. Glycol is blank, as water is reduced. Oxidized. Definition of enzyme activity. 1 unit of enzyme is the amount that catalyzes conversion of 1 micromole of substrate per minute, under specified conditions. What are the units for specific activity? E per milligram. What is rate law? Mathematical equation describing the progress of a reaction, contains a constant, K, which is Rate constant must be determined experimentally, cannot predict from a chemical equation, must use plots. What are the two forms of rate law for chemical kinetics? Differential rate law and integrated rate law. Define differential rate law. Relates the rate of reaction to the concentration of various species in the system. Define integrated rate law. Links concentrations of reactants or products with time. What are the orders of reactions? 0, first and second order. What the differential rate law for zero order? R equals d, A, dt equals K. What is the integrated rate law for zero order? A equals Kt, A, 0. What is the kinetic plot for zero order? A versus T. What is the slope for zero order? K. What is the differential rate law for first order? R equals d, A, dt equals K, A. What is the integrated rate law for first order? Lane A equals Kt lane A, 0. What is the kinetic plot for first order and its slope? Lane A versus K. What is the rate of reaction given by peroxidase activity? R equals d dialcohol, dt equals d tertiary glycol, dt. What is the rate of reaction given by blanching efficiency? R equals d E dt. All enzymes are proteins. RNAs with catalytic activity for synthesis and degradation of phosphodiester bonds. Ribozymes. Enzymes have an active site, which is polar in most cases, but its surroundings are vary. Hydrophobic. Some specific AAR chains actively participate in binding and transferring substrate into product, ES2, EP, 2EP. Enzymes are classified into blank classes based on the type of general reaction they catalyze. Enzymes affect blank not equilibria. Reaction rates. Enzymes are powerful and highly efficient catalysts. Enzymes can accelerate reactions in the range of blank to blank orders of magnitude. 5.17. The catalytic constant is equal to Kcat equals hashtag molecules S to P molecule enzyme X second. Some enzymes need essential cofactors such as metallic ions or coenzymes made from water-soluble vitamins and metabolites. Enzymes may transfer energy from one form to another. Most enzymes are not regulated and show simple behavior. A few enzymes are regulated and some show allosteric behavior. An allosteric enzyme catalyzed the key reaction in many metabolic pathways. The rate of enzyme catalyzed reaction is affected by several factors. Temperature. Too much temp denatures enzyme, pH, inhibitors and activators, switches. Ionic strength of medium, hydrophobic, lipid-like and cell membrane, character of medium. Rate is directly proportional to enzyme concentration. Trypsin pH is between 41,828. pH is between 41,602. Phosphates in body. Acidic and basic phosphates. Only enzyme W, no bell-shaped profile. Acetylcholinesterase. Naming enzymes, most common used is substrate ASE, sucrase, enzyme that hydrolyzes sucrose. Naming enzymes exceptions. Trypsin, a general protease and pepsin. Enzyme commission. EC 1-6. Enzyme class 1R. Redox. Enzyme class 6R. Ligases. Cofactors are required by inactive apid enzymes to convert them into active enzyme cofactor, enzyme cofactor, enzyme active. Two classes of cofactors: organic, inorganic.